Hello, I'm Rick Fair, President and CEO of Bellicum. It's great to be here with you virtually at the Cell and Gene meeting on the Mesa 2020. Today, I'm excited to talk to you about the progress we've made at Bellicum. I will be making forward-looking statements, and those statements have risks. I encourage you to read more about those risks in our SEC filings found on our website. Like many of the people at this meeting, Bellicum is really striving for a powerful new future in cellular immuno-oncology. We're doing that via our GOCAR platform, which has been engineered to enhance proliferation of immune cells, to enhance persistence by overcoming T-cell exhaustion and the immune suppression of the tumor microenvironment, by enhancing the power of these therapies, by engaging a host immune response, and ultimately to enhance the performance of these therapies by increasing control via our molecular switch platform. We've incorporated the GOCAR platform into 3 programs. Our first 2 programs are testing the hypothesis that we can establish greater clinical value in solid tumors. BPX-601 targeting PSCA in pancreatic and prostate cancers, and BPX-603 targeting a basket of HER2-positive cancers. Our third program tests the hypothesis that GOCAR can enable us to have a best-in-class allogeneic cell therapy. And our first program is a GOCAR-NK program targeting BCMA in preclinical development. I'll talk more about each of these programs here today. I'll start with our technology. GOCAR is differentiated from first and second generation CAR-T cells. On the left of this slide, you see the first generation, a CAR above the cell surface tethered to CD3 zeta signaling. We know from experience that this wasn't particularly active, and so the field added co-stimulation via CD28 or 4-1BB to enhance the proliferation and ultimately the efficacy of these CAR-T cells, leading to the current generation that's approved in some heme malignancies. GOCAR is different in a couple of important ways. First, we've selected different co-stimulation co-activation molecules, MyD88 and CD40, or what we call MC. We selected MC because it's really the most potent co-stimulation and proliferative signal we could find. Secondly, we've tethered that co-stimulation to our molecular switch system instead of being attached directly to the CAR. This system is based on science called chemically induced dimerization, or CID. We incorporate genetic modifications into the cells. They express these proteins, MyD88 CD40 molecules attached to FK binding proteins. These proteins, when expressed, do nothing in and of themselves, but once dimerized with our small molecule Remibrucid, they signal, leading to the co-stimulation that we're looking for. So our CARs are most proliferative and most activated when they see tumor antigen activating the first generation CAR and Remibrucid to activate MyD88 CD40 signaling. It's with this difference that we hopefully enhance the potency of these molecules and, of course, the controllability. I'll talk now about each of our programs, starting with BPX-601. As I mentioned, BPX-601 targets PSCA, or prostate stem cell antigen, which is expressed in a number of GI and GU tumors. We initiated a phase 1 dose escalation trial in pancreatic cancer and have now in the process of amending that trial to incorporate prostate cancer patients as well. We've presented a couple of updates on this study, and I'll share some of those data with you here today. We also expect to provide an update on the pancreatic cohort in the 4th quarter of this year. You see here the dose escalation from the trial to date. I share this as context for the data that I'll show. The trial was designed conservatively to ensure safety for a couple of reasons. First, PSCA is not an antigen that's been explored in CAR-T cell studies, and so we wanted to make sure that we were aware of any potential on-target off-tumor toxicity that could occur. And secondly, our MyD88 CD40 system has been demonstrated preclinically to be more potent than second-generation CAR-T cells. So we wanted to ensure that we could give that safely in human, as this is our first in-human experience. So with that conservatism, we added a few features. First, the lead-in cohort, Cohort 0. These patients received a low dose of CAR-T cells only without Remibrucid to activate them. So essentially a first-generation CAR-T cell. Through dose escalation, patients received only partial lymphodepleting chemotherapy with a single dose of Cytoxan. And we know from the CAR-T experience that full lymphodepletion, typically with a 3-day regimen of Flu/Cy, is associated with better engraftment expansion of cells and ultimately efficacy. The third and maybe most important conservative feature here is that through dose escalation, we only gave a single dose of Remidustat at day 7 to activate the cells once. IMC was designed to reactivate the cells over time, hopefully leading to improvements in proliferation, persistence, and activity. So it's really the Cohort 5C that we're currently enrolling, our first experience using weekly Remidustat dosing. It'll be our first test of that hypothesis. But through Cohort 5B, patients received a single dose of Remidustat, and these are the data that we've presented to date. I'll share some of that here. You see here the safety that we've reported through Cohort 5B. No dose-limiting toxicities have been observed. Adverse events were generally manageable and consistent with cytotoxic chemotherapy and the lymphodepletion regimen, as well as cancer immunotherapy studies. Adverse of interest associated potentially with BPX-601 and Remidustat, one case of grade 2 cytokine release syndrome, one case of grade 2 encephalopathy. Both of these occurred in Cohort 5B, and 4 cases of grade 1 to 3 urologic toxicity. We hypothesized that this urologic toxicity could be caused by the higher dose of Cytoxan being used in Cohort 5B in the Flu-Cy regimen. We know from the literature that Cytoxan is associated with bladder tox, and we also know that in CAR-T studies, patients generally benefit from prophylaxis for these adverse events. Starting in Cohort 5C, we converted those prophylaxis procedures to mandatory from optional. We'll look forward to seeing Cohort 5C and beyond at future updates to see if that has mitigated the bladder toxicity that we've observed. In spite of all of that, the adverse event profile looks favorable to date and allows us to continue in this dose escalation study. Given the conservatism of the design of this study at this time point, we didn't expect clinical activity, but we observed some modest level of biologic activity. You see here the swim plot of patients treated with cells and Remidustat up through Cohort 5B. 8 of 13 evaluable patients had achieved stable disease. 3 of those 13 patients achieved some level of tumor shrinkage between 10 and 24%. So not RECIST responses, but certainly some evidence of biologic activity. And we see several patients here with stable disease or treatment-free intervals out 6 months or more. Certainly not what we'd expect to see in a very late-line pancreatic cancer patient population, where progression-free survival is something more like 8 to 12 weeks. So perhaps some evidence that we're having some biologic effect in these patients with this very conservative dose and schedule early in this study. BPX-601 is important to us, not just because of the unmet need in pancreatic cancer and prostate cancer, but it's also our first in human experience with GOCAR-T cells, and we certainly want to learn about the performance of the platform in humans. We'll look first here at proliferation and persistence as measured by circulating BPX-601 cells by vector copy number. You see here all of the patients in the study treated through Cohort 5B aggregated by cohort. The black line along the bottom of this graph is Cohort 0. This was the cells-only lead-in cohort, a low dose of cells. You see what you'd expect to see in a first-generation CAR-T cell study in solid tumors. Very little proliferation, a peak vector copy number of about 1,000 copies, very little proliferation. Cells were detectable in the periphery beyond 3 weeks. We've enhanced the proliferation and persistence with each step of the study to date. You see the green line across the top is Cohort 5B. These are patients who received 5 million cells per kilogram after receiving Flu-Cy lymphodepletion and then received a single dose of Remidustat at day 7. And what you see are very nice levels of proliferation, up to about 20,000 copies per microgram, and persistence out 6 months or more. These results compare very favorably to what's been reported for second-generation CAR-T cells in solid tumor studies. So we feel good that IMC is having some positive benefit in terms of proliferation and persistence. We're also interested in making sure that the system's working as intended, that we're actually able to activate CAR-T cells with our small molecule CID system. You see here circulating cytokine levels over time, starting at day 0 when the cells were infused. You see a peak from the initial expansion of those CAR-T cells into the lymphodepleted T cell compartment with a spike in those cytokine levels, largely pro-inflammatory cytokines at around day 4. Returning to a much lower level of cytokine production by day 7. On day 7, these patients received a single dose of Remidusib, and you see by day 8, again, upregulation of a bolus of pro-inflammatory cytokines. This is certainly evidence of CAR T cell activation with our small molecule system, and also part of our hypothesis about the benefit of IMC, that we will be able to create inflammation with our system that can help contribute to the immune response in these tumors. One of the questions about CAR T cells in solid tumor studies, of course, is can they get there? And we have evidence from a small number of patients from Cohort 5B, where we were able to gather pre and post treatment tumor biopsies in a handful of patients. You see here the 3 patients who had pre and post treatment biopsies, and you see evidence of BPX601 infiltration in all 3. You see a couple of the slides here with demarcated BPX601 cells, generally localized to tumor cells. So a good sign that at very minimum, our CAR T cells are trafficking to these lesions and could potentially have a biologic effect. One of the last translational items we looked at was really evidence of inflammation in the tumor microenvironment, again, leveraging those same 3 patient biopsies that I described on the previous slide. And very importantly and nicely, we see pretty consistent upregulation of genes associated with immunity and T cell immune responses, granzyme B, CXCR3, 4-1BB, and others. Certainly evidence to us that we are tipping the balance in the tumor microenvironment towards inflammation, and that there may be a possible benefit from that as we see the host immune response contribute to anti-tumor effect. So good evidence so far that the GOCAR system is working as we intended, evidence of biologic activity in BPX601 patients, and evidence of good safety. We'll look forward to the next step in this study, where we're enrolling patients receiving weekly Remidusib doses to reactivate cells, and to continuing the study moving forward. Turning now to BPX603, this is our dual switch GOCAR-T cell targeting HER2. Dual switch is nomenclature for incorporation of not just our IMC activation switch, but our caspicide safety switch, which has been used in clinical experience to eliminate genetically modified cells via apoptosis with the same chemically induced dimerization system and a second small molecule. So it really enhances our ability to manage safety events should they occur. Obviously, HER2 is a great place for us to test the potential benefit of GOCAR-T cells. It's a well-validated cancer target, and there has been quite a bit of academic experience with CAR T cells targeting HER2, so we have a bit of a historic control of second-generation CAR T cells in this setting. We've enhanced the design of this to try and enhance the efficacy that has been observed. The efficacy observed in academic trials has been roughly an 8 to 10% overall response rate, so mildly active, and we certainly know we need to enhance that to have a product. We've done that in a few ways. We've increased the affinity to a sort of a moderate affinity of the binder single chain variable fragment to enhance target engagement. We've of course incorporated our IMC activation switch, and of course we've also incorporated the safety switch, which gives us an additional safety margin to be a little bit more aggressive. The IND for this project has been cleared. We are in the study startup for a phase 1/2 study and expect to initiate that trial this year. You see here the trial design. It's a phase 1/2 study. Phase 1, largely classic 3+3 dose escalation through at least 4 dose cohorts. These patients will be enrolled sequentially with a 28-day wait in the first dose level and 14-day wait in subsequent dose levels. Interestingly, one modification that we've made just to ensure safety of these patients in the 3+3 is that the first patient at each dose level will receive CAR T cells alone without Remidusib to activate them. And then the second and third patient in each cohort will receive cells followed by weekly doses of Remidusib to either disease progression or dose-limiting toxicity. So it gives us an opportunity to ensure the CAR itself is safe at each dose level before we activate the system. Once we have a phase 2 dose, we'll move into a multi-arm dose expansion with small expansion cohorts and a number of HER2-positive tumors, and ability to expand those cohorts based on data as we move forward. As mentioned, the study will start later this year. We'll look forward to presenting results hopefully sometime late next year, first results from dose escalation. Finally, turning to our earlier preclinical GOCAR-NK program, we set out to find a pathway to an allogeneic cell therapy leveraging GOCAR and looked at a number of cell types. We really like what we saw with NK cells. Of course, NK cells are an attractive area for many, and many are pursuing them. They have an innate ability to kill tumor cells by themselves. They also have a favorable safety profile when used in adoptive cell transfer. They don't cause GVHD in the same way that T cells do. So there's a lot to like about them as a platform. Of course, they've been limited previously, largely due to poor in vivo expansion and persistence. And we believe that our system gives us an opportunity to improve that. Over the last 18 months or so, we've done quite a bit of discovery work with GOCAR in NK cells, and we've presented that on a couple of occasions and have published those data recently in Blood Advances in May. To summarize the findings from our discovery work, we see that MC signaling, particularly when combined with IL-15, where we see synergy, improves the proliferation and survival of NK cells in vivo. That's an exciting finding and difficult to achieve. We also see that MC signaling enhances the innate cytotoxicity of these NK cells. And we think that innate cytotoxicity is really important. No single tumor antigen is going to be perfect. And so CAR-directed killing is only a part of the story. And certainly the idea of having, say, supercharged NK cells to clean up residual tumor cells that may have low or no expression of the CAR target is an attractive feature here, and one that we think our system enhances. So we've looked at multiple in vivo tumor models of IMC IL-15 enabled GOCAR-NK cells and have seen superior anti-tumor effects in all of them. So based on that preclinical work, we've advanced to candidate selection, a BCMA GOCAR-NK program. Obviously, BCMA is an exciting target. We know that cell therapy will play an important role in multiple myeloma treatment based on the results that have been achieved with autologous CAR T cells. We think that, of course, allogeneic T cells or allogeneic cell therapies, T or NK, have advantages of ability to manufacture at lower cost, quicker and more reliable access to treatment for patients. And so if an allogeneic cell therapy can show comparable clinical outcomes in a more convenient package, that's going to be super attractive. Based on our preclinical work, we feel very confident that a GOCAR-NK cell has a chance to be a best-in-class allogeneic cell therapy in the space. And so we are moving forward now to finalize the construct design and final candidate selection to take this product into the clinic. Our timelines really are about finalizing the candidate this year and finalizing our manufacturing methods. We've developed some novel feeder-free NK cell manufacturing methods around this program that we'll be optimizing and scaling this year. Next year, we'll be very focused on the IND enabling studies such that we can file and clear an IND hopefully in the early part of 2022. You see here our milestones. I've talked about many of the near-term milestones. We're very excited about data events later this year. BPX-601 Cohort 5C first data in pancreatic cancer. Also, we'll present a preclinical data update on our BCMA program at a medical meeting in the fourth quarter as well. And then we see really an accelerating data flow from these three programs as we play out the GOCAR platform in each of these two different applications to see what we see and see if we're contributing enhanced efficacy and clinical benefit for these patients. In closing, we're excited about our platform. We think it offers the ability to drive proliferation, persistence, power, and performance in T and NK cells. We've applied that to three really exciting, differentiated, and wholly owned programs, and we'll look forward to keeping you updated in the next year or two ahead as these programs advance to proof of concept. Thanks very much for your time today, and enjoy the meeting.